we're going to dive into the actual methods of control that are used. And they're divided into two categories. Physical methods, which would include things like heat, irradiation, filtration, and just mechanical removal like hand washing. And then in the next video, we'll talk about some chemical methods of control. So these would be all the chemicals like betadine, or hand sanitizer, or bleach spray, or Lysol, all the chemicals that are used to control microorganisms. So we'll start off by talking about the physical control methods we said in this video. And so heat is probably the most common, but we can also use filtration, radiation, and we're going to talk about high pressure, which kind of goes into heat. It feeds into that. This table just summarizes some of the key points for the control methods, but we'll go over them in more depth as we go through this video. So we start off with heat. Usually heat is the most useful for microbial control because it comes with a lot of advantages. It's pretty fast. It doesn't take a long time for heat to kill a microbe. It's reliable. They don't really develop resistance against heat like they do to chemicals. It's pretty safe as long as you're protecting yourself and you're not burning yourself. It doesn't come with things like toxicity. And it's inexpensive. You can generate heat multitude of ways that are really cost efficient. So depending on how the heat is used, it can be used to sterilize or disinfect. And there's actually two types of heat. There's moist heat and dry heat, and both have a place in microbial control. So moist heat would be like boiling or pasteurization. So boiling, that would be increasing the temperature above 100 degrees Celsius. And that's typically sufficient enough to kill most microorganisms and viruses. But we know that it's not a means of sterilization because it doesn't kill endospores. Endospores can survive boiling. So it's not completely removing every single microbe, but it can substantially reduce the microbial population. We also know that temperature to which water boils increases as you increase elevation. So in certain places with higher elevation, the temperature is a little bit higher. But typically, boiling water at 100 degrees Celsius for 5 minutes is enough time to kill most of the microbes in the water. And the CDC actually says that in 1 minute, that's enough time to destroy all of the vegetative cells and the enveloped viruses in that water, or whatever it is you're boiling. Pasteurization. Now, we said pasteurization is brief heat treatment. And so pasteurization can be used in two different ways. It can be used to provide a really short burst of high heat for like liquids or food products, but it also can be used to disinfect like containers, like milk jugs and things like that. And so it's really just flash heating the item or the food. And so by doing this, you're instantly killing most of those microbes because heat works pretty fast, but you're still keeping the integrity of the food product. There's two protocols that we use for pasteurization: high temperature, short time, and ultra high temperature. In high temperature, short time, let's say milk, is pasteurized this way. It runs through a series of tubes, and when it hits the warming section of the tube, it will rapidly heat the milk to 72 degrees Celsius for 15 seconds. And then it immediately passes into a cooling tube where it then cools it down. So it's very brief flash heat so that the proteins and stuff in the milk and the taste, it still remains the same roughly. There are people who say that they can tell a difference in the taste between pasteurized and unpasteurized milk, and there are some proteins that do get broken down through pasteurization, but for the most part, the integrity of the milk stays the same. Ultra high temperature, that's going to be 140 to 150 degrees Celsius, so significantly hotter, but for a shorter amount of time, only for 3 seconds. So this is used for a lot of products that get packaged and sealed so they can be stored at room temperature. So if you've ever seen like milk packets or fruit juices, some soups, even honey can be pasteurized this way. It's used for those coffee creamers. If you've ever wondered how a coffee creamer can sit out at room temperature and not spoil, it's because it's been ultra high temperature pasteurized. There's also something you may have heard of called vat pasteurization. It's a little bit different protocol. It's 155 degrees Fahrenheit for 30 minutes. So it's a lower temperature and a longer time. And sometimes they do that for milk because it will not cause that breakdown of proteins in the milk. So some people use it because it will make the milk more nutritious. So you may have heard of vat pasteurization. But high temperature short time is faster, it's more energy efficient. Even though it does give a slightly cooked flavor, it is the preferred method for milk pasteurization for the milk you buy at the grocery store. We also use pressurized steam. This is where autoclaves come into play. So an autoclave is basically a big chamber that will increase the pressure and temperature simultaneously. And what we know is that steam will form at higher temperatures under higher pressures. So we get really high temperature steam, and that can be used to sterilize. So typically everything that we work with in the laboratory has gone into the autoclave. You can see it's got this big chamber. You put your glassware, your utensils, your media, so like all the media we use in the petri dishes and all of that, that goes into the autoclave. It has an airtight seal on the door. You push a button, it slowly increases the pressure to 15 pounds per square inch. That's what 15 psi is. And it increases the temperature to 121 degrees Celsius for 15 minutes. And that's enough to kill everything, even endospores. The only thing that it can't kill are prions. If you want to work with prions in the laboratory, they actually have to increase the temperature to 132 degrees Celsius for 4.5 hours in order to kill the prions, because prions are so heat stable. So typically 15 minutes is enough unless you are a very special lab that works with prions. So those are examples of moist heat. Next we look at dry heat. So it's not as effective as that steam that's created in moist heat. So usually the sterilization times take longer and they have to be done at higher temperatures. But incineration is our most common method for this. So anytime that you flame your inoculating loop, you're using dry heat when we're in the laboratory, when we're transferring bacteria. Also medical and animal waste, medical waste and animal carcasses in research facilities, they would be incinerated. We can use something called gas sterilization, in which we use high heat and gases in order to sterilize that item. So they can be, we can do a physical and chemical control method together. Sometimes that happens. You can see in the picture here what's that wrapping, that blue paper, that's like autoclave paper. And so it's heat stable paper, so you could put like a whole bunch of needles in there or whatever you want to work with. You can wrap it in this heat stable wrapping, tape it. That's even special tape on there that turns black when it's reached a high enough temperature. Those black lines form, so you can see on the left hand side, those they look like strike marks through the tape there. That's to show, yes, the autoclave worked. It reached or not autoclave, but the chamber worked. It reached a high enough temperature and it has been sterilized. So gas sterilization, things like examples would be like ethylene oxide. That's commonly used to control microbes. Next, the microbes can be filtered. So there are some liquids that just can't survive heat. Like glucose kind of falls apart. It gets all yellow and gross when it's been autoclaved. So for those types of liquids, we would use filtration. And so we just use membrane filters, and so they're porous filters, and the pores are large enough to allow the liquid through, but the microbes get trapped on the other side of the filter. So you can see the apparatus here. It's that we use a vacuum, and so we have a flask, and then we have our funnel. We pour the liquid into it, and then the vacuum will pull the liquid through the filter, so we don't have to wait for it to titrate naturally, because it would take hours. We suck it right down with the vacuum, and all of the microbes get trapped on the other end of the filter. We also can filter air as well, and the HEPA filters are really good for filtering air. It stands for high efficiency particulate air. And we can use HEPA filters in microbiology hoods. They're biological hoods. It's different than a chemical hood. In a biological hood, it's not attached to the wall and hooked up to the main ventilation of the building. It's actually transportable, and you create a sterile environment. You know, you spray it down, you wipe it down, and then it actually sucks in the air and up out the HEPA filter and just dumps the air right back on top of you. Whereas a chemical hood, the chemical fumes obviously get sucked up and out the building. So biological hoods that we use in microbiology labs, they have HEPA filters. They can also be used in facial respirators, so you can filter the air directly that you're breathing. You probably have seen pictures of people wearing gas masks, right? They have the window for viewing, and then they have the two little circles sticking out of them. Those are the HEPA filters. That's what actually filters the air. The last type of physical control we're going to talk about is radiation. So there's two types of radiation, and they both kill microbes, but they do it in different ways. Ionizing radiation would be things like gamma rays, X-rays, and electron accelerators. Non-ionizing radiation would be like ultraviolet light. Now, they both damage DNA, but how they damage the DNA is different. So we're going to talk about modes of action. And for whatever reason, this tends to be the most difficult part of this chapter for students to comprehend. Ionizing radiation breaks chemical bonds. Non-ionizing radiation causes abnormal bonds to form. So they're really doing the opposite thing. So ionizing radiation causes ions to form. And those ions form when electrons get ejected out of orbit. So they get really excited, and that ionizing radiation causes them to shoot out of their orbit. Electrons are responsible for chemical bonding. So bonding between two atoms happens with the electrons. If the electrons are ejected out of orbit, that bond breaks. Now, when that happens to the DNA, the cell dies. The cell cannot survive if its DNA is broken. Remember, the DNA is the blueprint of the cell. Ionizing radiation has pretty good penetration power, can get through most barriers. That's why you have to wear a lead vest in certain places. Like if you're getting dental x-rays, you wear a lead vest. If you're getting x-rays in other parts of your body, they cover it. The areas not getting the x-ray with the lead blanket. Non-ionizing radiation is different. Non-ionizing radiation causes formations of something called dimers. These dimers occur, so dimer means two of the same molecules bonded together. So we have to think about the structure of DNA. DNA, there are two complementary strands of DNA bonded to each other, and the A nucleotides always bind to the T's, and the C nucleotides always bind to the G's, right? There's 4 nucleotides in DNA, A, T, C, and G. When you shine UV light on cells, the bonds between two of the same nucleotides form. And so you get thymine dimers, 2 T's bound together, or you get 2 C's bound together. And those abnormal bonds are like taking a string, if your DNA is the string, and just knotting it all up into a big ball, and it's all tangled. If it's all tangled up, the RNA polymerase can't read it and make messenger RNA from the DNA. And then proteins never get made and the cell dies, because proteins control everything. Without their DNA, they cannot survive. Now, we do know that non-ionizing radiation, UV light, has really poor penetrating power. It cannot penetrate through barriers, even if they're transparent. So as we'll see in the lab, the Petri dishes that we use on top of our media plates, that clear plastic Petri dish is enough to block the UV radiation. So even though it's great, and I have seen some like daycare facilities use it, like after the students leave, they turn on the UV lights and it will help disinfect surfaces and stuff. Because of its low penetrating power, it does have limited use. If we go back to how they are used, so we said ultraviolet light can be used for surfaces and things like that. It can be used to disinfect drinking water. So they make UV light sticks if you're somebody who does a lot of like outdoor backpacking and you're not bringing your own water and you're going to be drinking water on site. They're just little sticks you stick into, your Nalgene. After you pick up your stream water, you push a button and it shines UV light into the container and kills the microbes. Ionizing radiation, we talked about how gamma rays can be used in the food industry for really delicate things like spices. They're used for medical supplies. They can use it for mail when they're feeding mail through the machines. After we had the big anthrax scare in the early 2000s, which is a bacterial infection, Bacillus anthracis that causes anthrax, there's a risk. Mail's handled by lots of different people, and so it's full of microbes. And pharmaceuticals as well sometimes get gamma radiation. So it is used, it's just probably not as commonly known as ultraviolet light is. Alright, so those are our physical methods of control. In the next video, we're going to begin to talk about chemical methods of control.